Well, thank you very much. My name is Asaf, and I'm the CEO and co-founder of therapeutics, and I'm from Tel Aviv, Israel. Basically, what we do is we print organs. How do we do it? Some background about tissue engineering. Usually in tissue engineering, we take cells from the body, we cultivate them on two-dimensional cultures, move them to 3D scaffolding, and then re-implement them into the body. Well, there are many companies that are doing tissue engineering, but usually they use cells that are from the patient, but the matrix is either from other people, from animals, or synthetic. And therefore you have an immune response that is damaging this implant. What we did is we are using patient-specific cells and matrix. We use the patient's own cells and matrix. We produce it very easily from the fatty tissue, and then we create a thermoresponsive hydrogel, and within this gel, we do the differentiation to each and every tissue, like you see in this schematic drawing. Our work was published in numerous leading articles in the world, including Nature, Nanotechnology, Biotechnology Engineering, and more. And I would like to share with you some of our results in the preclinical era. What you see here is a myocardial infarction. Okay? An artery is occluded, there is no blood supply, there is no oxygen supply to the tissue, and the tissue simply dies. What we did in the lab is we created a heart tissue. We created a patch that we can take this patch and basically put it on the heart. Okay? After we put it on the heart, it goes complete integration into the heart muscle and start to work as the patient's own heart. The problem with this procedure is that you need to open the chest. You need to do a surgery to open the chest and put the patch on the heart, like you see in this movie. Okay, this is the heart with the patch on it. So what we did next is we created a gel that is liquid at room temperature, and when it gets into the body, it turns into a sticky gel. So now what we can do is basically inject it with a syringe, like you see in this movie. We take a syringe and inject the gel into the patient. But now when we have the gel, we can do other stuff with it. We can, for example, take the heart, take only the blood vessel, create a plan to the printer, and send it to the printer. And then the printer basically prints blood vessels, like you see in this movie. Okay, it takes the plan that we send the printer and prints blood vessels. And we can pick this patch, and when we inject blood into it, you see that the blood is flowing through the blood vessels. And in order to prove that these are blood vessels, you can see in this movie that it has fibroblast and endothelial cells. So it is a blood vessel. So the next phase was done in April. We printed a whole heart. The first ever in history. A printed human heart. And this got a lot of news all over the world because this, what I hold in my hand, is history. And we are going to change history because we offer not only printing of hearts, we offer printing of many other organs. For example, what we are working now is on spinal cord. And you can see here, the movie doesn't work, but you see that we injected spinal cord that we created in the lab to mice that had a spinal cord injury. They were paralyzed. And you see over here that in the group that we didn't do anything, we just cut their spinal cord, you see that the mice is limping and he does not use one of his legs. When we inserted the gel, only the gel without cells, 70% of the mice started walking again. And when we added motor neurons, 100% of the mice regained their walk, started walking again. We are planning to do, within 2.5 years, the first clinical trial in humans for spinal cord injury. And just try to imagine that out of the 10 patients in the study, one patient will move his finger. This will change medicine forever. We have a great team of researchers, physicians, and support team working with us on this amazing project. And we believe the future is here. Thank you very much.
Thank you, Asaf. 很高兴我们今天一起在这里见证了这么多激动人心的项目。想听听4位老师有什么样的问题？郑老师，
Wonderful job. So I think your cells you use is reprogrammed from the somatic cells and then differentiate or undifferentiated.
We take a omental biopsy from the omentum, and then we take the matrix and do a different method for the matrix and different method for the cells. We create IPS, and then we combine them together inside the gel, and the actual differentiation to the certain tissue is being done within the gel. So the cells are being connected in the gel, so when we inject it, they are already connected as a tissue.
Yeah, for the IPS, since 2006, it's being developed by the Japanese scientists. The most problem is that whether it is safe to be used in human. So what's your consideration? And my final question is another one is that you have done a wonderful job. You print 3D heart, and I want to know which kind of problem you think is the biggest problem in the future. I think, in your opinion.
So first, I'll go with the IPS. There are currently methods of messenger RNA that are much more safer and are already in clinical trials in the world. So it's not a problem like it was in 2006 when it was just discovered. The main problem that we have currently in the lab is how to train the heart to function as a heart. We know how to print it, but now what we are doing in the lab is actually train the heart to function as a heart, to contract and to send blood. And this is actually what we are doing now in the lab. And the next animal study will be to implement this heart next to the heart that is functioning to see that the circulation works and the heart itself works. And then the next phase will be to implement only our heart to replace the heart.
More question?
So at the end of your presentation, you mentioned you're planning to start clinical studies on spinal cord injury.
Yes.
So my question is, do you see that as a faster entry point into the market compared to
Definitely.
the heart, right?
Definitely, because regulatory-wise, it's a compassionate treatment. So those patients have no other option of treatment.
Absolutely.
So this is why regulatory-wise, FDA, CFDA, the European Union, it's much easier to go for the spinal cord injury than the heart. But, as I was written on the presentation, The next phase, the heart, will be easier because all of the safety features and most of the preclinical studies will be acceptable for the heart as well.
Right. Absolutely. So I kind of look at it like similar to Elon Musk's strategy with SpaceX, right?
Yeah.
So it's a long road to go to Mars, but in the interim, you build up your capability on sending commercial rockets into space. So this is kind of similar to that strategy.
Both of us from Tel Aviv.
Okay. No wonder. Thank you.
Okay, thank you.
Thank you very much.
Thank you, Yosef.